The Chinese boasted of their successful experiment in a scientific journal, but Western scientists were shocked by this research. In a Chinese laboratory, scientists, for the sake of an experiment, created a mutated strain of COVID-19, which showed 100% lethality in mice whose genes were as close as possible to human ones. This is stated in the research materials released last week in Beijing. The GX-P2V virus is a mutated version of the GX-2017 virus, which is called the cousin of the coronavirus as we know it. During the experiment, the researchers used laboratory mice whose genes were engineered to be similar to those of humans. All mice infected with the GX-P2V virus died in just 8 days, which the researchers noted was a surprisingly rapid mortality rate. The virus infected the lungs, bones, eyes, tracheas, and brains of mice. The latter was serious enough to eventually lead to the death of the animals. In the days before death, the mice rapidly lost weight, had a hunched posture, and moved extremely sluggishly. The worst thing is that their eyes turned completely white the day before they died. This highlights the risk of GX-PV spreading to humans and provides a unique model for understanding the pathogenic mechanisms of viruses associated with SARS-CoV-2, the authors write. François Balloux, an epidemiology expert at the Institute of Genetics at University College London, called the study horrible and completely scientifically meaningless. I don't see anything interesting that could be learned by forcibly infecting a strange breed of humanized mice with a random virus. Conversely, I have seen how many things can go wrong, the professor wrote on Twitter. He noted that the summary report of the study's results, which was made public, did not indicate what safety measures were taken during the experiment. The lack of this information raises concerns that some or all of this research, such as the 2016-2019 Wuhan study, that likely caused the COVID-19 pandemic, was recklessly conducted without the minimum biosafety conditions and practices required for research with potential pathogens of the pandemic, he noted.